Hi everybody, this is Dr. Launderville. I want to talk to you about the hypotheses. I just returned them to you and I'm not going to record those grades. Instead, I'm going to give you another shot at doing the hypothesis. This is going to be hypothesis take 2. There's a folder available in the assignment folder for hypothesis take 2. That one is going to be due on Sunday the 21st. Which is a week from today. And you're going to take my feedback from your first shot at doing a hypothesis, plus do some more preparation, and then take another shot at doing the hypothesis, because it was obvious that you didn't get enough direction from me on the first time because nobody really got it. So for your take 2, I want to make sure that everybody uses the language that says, I will test the hypothesis that, I will test the hypothesis that, and then you write down your hypothesis. You must use that language. Almost all of you wrote the species name wrong. It is C. elegans with both the C and the elegans in italics. It is not C. elegans not in italics, or only elegans italicized, or elegans with a capital E, or elegance, that's a different word, etc. It is C. elegans, both the C and the elegans italicized. Okay. Please read the guidelines that I have posted. So I have posted guidelines now for the entire proposal. There are many common mistakes listed in those guidelines. Many of you are making those common writing mistakes just in the one sentence hypothesis. So I want you to take a look at that before you take your second shot at writing the hypothesis. I also want you to watch the 3 videos about how to write a proposal. You're not writing the proposal at this stage. You're just writing the hypothesis at this stage. But some of you don't really know what's ahead of you in terms of writing the proposal, and therefore you're proposing to do things in your hypothesis that can't be done. So, for example, proposing things that can't be tested. So in your hypothesis, at the end of doing the experiment, you should have data that either support your hypothesis or don't support your hypothesis. If you're proposing something that is so vague and not precise enough that you can't really say that you did or did not support your hypothesis, then it's not a good hypothesis. So I want you to watch all 3 videos. You would be watching those anyway. Before you write your next hypothesis. Those videos are on how to write the proposal, but they have to do with the hypothesis. Also want you to read the guidelines for the proposal, which are about the proposal, but includes common writing mistakes that many of you are making in writing the hypothesis. Okay, it was obvious that many of you just whipped this off and didn't do any background reading before you wrote your hypothesis. I want you to be thinking when you're writing your hypothesis, what will we learn as a result of doing this experiment? So, for example, many of you, maybe a dozen of you, had hypotheses that had to do with light exposure and C. elegans. You have to realize that C. elegans lives in the soil. You should know that from your background reading in the Wormerama lab, and therefore they're not exposed to light. So it doesn't mean they don't have a response to light, but it does mean that how we interpret their response to light is different than an organism that lives in light. So light is a stressor for these animals, and you have to think along those lines if you are proposing a hypothesis that has to do with light. And along those lines, I don't want you to see hypothesis that has just some new way to stress out worms and then a test to see did it really stress out worms. We know the answer to that question. We know what stresses out worms, and we're not interested, I'm not interested in finding out new ways to stress out worms. I'm interested in finding out new ways to learn things about these organisms and how they live, in a cell physiological context. So I want you to focus on cell pathways. I want you to focus on assays that you could do in a cell physiology lab. And if you want to measure something like the effect on fecundity or the effect on longevity, those are fine. But when you get to the proposal stage, you're going to have some other sort of cell phys assay that you describe. It can't be that you're just counting embryos for your experiment. You're going to need some kind of blot, some kind of qPCR, some kind of enzyme assay, something that we can do in the lab that pertains to cell physiology, not just counting things under a microscope. You must include a background paragraph. I will not grade these background paragraphs, but you have to give me something that goes to your thinking as to why you're asking this hypothesis. So some kind of rationale. It does not have to include citations, but if you did read, and you should read before you take another shot at this hypothesis, you should tell me what you read and what you know and why you are asking this question. Okay? For some of the ones I read, it was obvious that people had not done that, and I said so. For some of them that I had read, it was obvious that people had not looked in the primary literature to see if somebody else had done them, because 30 seconds on Google and I found out somebody has already done it. So you should do that work before you turn in your second chance at the hypothesis. Has this been done before? Is this an open question or not? Only the ones that are open questions are the ones that we're interested in. Okay, so once again, next Sunday, the 21st, you're going to take another shot at hypothesis. You're going to watch the 3 videos before you do that. You're going to read the guidelines. You're going to do some background reading about C. elegans and the particular topic you're interested in to find out what has been done and what hasn't been done, what is still an open question. You're going to write a specific hypothesis that is not general and vague and uses the correct language that says, I will test the hypothesis that. And then you're going to think about the significance, the novelty, and the writing. That's in the rubric. So significance means if we could do this experiment, would you make a contribution to what we know about C. elegans or what we know about cells in general? The novelty is, has this been done before? Has this approach been used before? Okay. Along those lines, a lot of people were, I'm going to knock out this gene or I'm going to knock down this gene and then I'm going to test whether or not it's knocked down. That is not novel. We know that the gene knockdown technology works, and so I won't advance any hypothesis that use that sort of approach. Okay? It has to be something like, here is what we know about when these genes are knocked down, but here's something we don't know about an effect of this knockdown, and I'm going to test that. That's the sort of thing that you want to go for. Okay? Look forward to seeing your second round of hypotheses.